and welcome. I'm Kelly Gannon, the Northeast Academic Account Manager at Cell Signaling Technology, or CST, and your moderator for today's webinar, Chromatin Profiling Just Got Faster with Cut and Tag. Our speaker today is Dr. Fong Chen, Associate Director of Epigenetic Applications at CST. She has been with CST for almost 8 years, holds a PhD from the Chinese Academy of Sciences, and completed her postdoctoral research at Yale University. She has over 10 years of expertise studying the interactions between proteins and DNA in various models such as cancer cells, animal tissues, and plant tissues. Since joining CST, she has been actively involved in the development and validation of several epigenetic assay kits, including the Cut and Tag kit, the Cut and Run assay kit, the Sonicase and Chip assay kit, and DNA library prep kits for next-gen sequencing. She and her team are responsible for validating all CST antibodies for Cut and Tag, Cut and Run, ChIP-seq, and ChIP applications. Dr. Chen also interacts with customers on a daily basis and has amassed an extensive experience addressing experimental pain points and providing technical support to researchers performing chromatin profiling experiments around the globe. Before we begin, I would like to briefly introduce CST. In a category where publicly traded multinational life science companies predominate, cell signaling technology is an outlier. CST is a company founded, owned, and run by active research scientists, people dedicated to helping unlock the secrets of life. Our mission is to empower the global scientific community, accelerate biological understanding, and enable personalized medicine, all with the highest standards of product and service quality, technological innovation, and scientific rigor. Today's webinar on Cut and Tag will conclude with a live Q&A. We invite you to ask questions by typing them into the questions area on your GoToWebinar control panel, both during the presentation and during the Q&A portion of the webinar. And now, without further ado, I'll turn it over to our chromatin profiling expert, Dr. Fong Chen.
Thank you for your introduction, Kelly. Good afternoon, everyone. As Kelly mentioned, there have been significant advancements in epigenetic applications over the past few years, transitioning from ChIP-seq to Cut and Run and now to Cut and Tag. Today, we will look into the workflow of the Cut and Tag assay, comparing it to Cut and Run regarding their respective protocols and benefits. We will discuss which assay aligns better with your experiment's needs. I will introduce all of the available CST kits, reagents, and antibodies validated for the Cut and Tag assay, along with the designs behind them. If we are following an in-house protocol, you will have the opportunity to select the most suitable reagents and antibodies following my introduction. Alternatively, if you prefer a commercialized kit, you will gain insight into our intensive validation work and the expected results from the CST Cut and Tag assay kit. Additionally, I will highlight multiple Cut and Tag resources we offer and address some of the most frequently asked questions. If you have any questions during our session today, I will gladly provide answers. If in case the time doesn't permit, I will follow up via email to address any concerns. So now let's get started. Before the discussion on Cut and Tag, I'd like first to introduce the different classes of target proteins in chromatin profiling assays. Different target types present varying levels of difficulty in chromatin profiling assays. This is true for all of the ChIP-seq, Cut and Run, and the Cut and Tag assays. The first target type is histone modifications, which are considered the easiest targets because histone have very stable binding to DNA, and there are a large number of histones per DNA fragment. The second target type are transcription factors, which pose more challenges compared to histone modifications because they are less stable binding with DNA than histones. Also, there are fewer factors bound per DNA fragment. Finally, cofactors represent the most challenging target type because they often do not directly contact DNA. They just associate with DNA through protein-protein interactions, and they are often involved in large protein complexes, so their antibody epitopes can be easily buried. With this understanding, we'll be mindful of all target types as we introduce the CAT-and-TAG assay or compare it with other assays today. CAT-and-TAG stands for Cleavage and Targets and Tegmentation, which was also developed by Dr. Henikoff's lab. It can be thought as a sister technology to CAT-and-RUN to look at protein-DNA interactions. Similar to CAT-and-TAG, similar to CAT-and-RUN, CAT-and-TAG also utilizes target-specific primary antibody, but it uses PAT-TN5 instead of PAT micrococoa nucleus to isolate protein DNA complexes. At the beginning of the experiment, cells are immobilized on concanavalin A magnetic beads to allow for subsequent buffer and reagent exchanges. Then, cell membranes are permeabilized with digitonin to facilitate the entry of antibody and enzyme into nuclei. Once inside the nuclei, the target-specific antibody seeks out and binds to the chromatin bound target protein. These three steps are the same as in the CAT-and-RUN assay. Next step, a secondary antibody is added, as CAT-and-TAG requires amplification on signals. The use of a secondary antibody is unique to the CAT-and-TAG assay and not to the CAT-and-RUN. While we occasionally find that the secondary antibody is optional with strong primary antibodies, for the majority of primary antibodies, incorporating a secondary antibody aids in CAT-and-TAG signal amplification. Therefore, we have standardized the use of the secondary antibody for protocol consistency. After that, the PAT-TN5 fusion transposase is recruited to the chromatin through the protein-protein interactions between the antibodies and the protein A protein G domain of the fusion enzyme. While this step is similar to that in CAT-and-RUN, CAT-and-TAG employs a distinct key enzyme, the transposase TN5, instead of micrococoa nucleus. The PAT-TN5 enzyme is preloaded with adapted DNA before being added into the reaction. Once activated by mechanism, the TN5 enzyme conducts tegmentation in CAT-and-TAG assay, cutting tegmented genomic DNA and ligating adapted DNA on site. This step differs significantly from CAT-and-RUN, where DNA is solely digested and an in vivo adapt ligation step is required during NGS library prep for CAT-and-RUN samples. However, in CAT-and-TAG, both DNA digestion and adapt ligation occur in vivo by TN5, saving time for subsequent NGS library preparation. Another notable difference in CAT-and-TAG is that the sample is incubated with SDS at 58 degrees to break open membranes and to ensure the solubility of the tegmented DNA, resulting in the final CAT-and-TAG DNA sample being composed of both target-bound tegmented DNA and background genomic DNA. Consequently, CAT-and-TAG DNA samples are not compatible with qPCR detection, unlike CAT-and-RUN DNA samples, which can be detected by both qPCR and NGS. Following tegmentation, CAT-and-TAG DNA is purified using DNA purification spin columns, just like CAT-and-RUN. The small adapt bound desired DNA is then selectively amplified by PCR, making it ready for NGS. As mentioned, the library prep procedure for that CAT-and-TAG assay is highly streamlined. After understanding the CAT-and-TAG workflow, we can grasp the benefits it offers. Similar to CAT-and-RUN, CAT-and-TAG has low sample requirement. We recommend starting with 100,000 cells, though it can be as low as 5,000 to 10,000 cells for histone targets and 20,000 cells for non-histone targets. This is 40 times fewer cells than required for the CHIP assay. For tissue samples, CAT-and-TAG can start with as little as 1 mg of tissues, which is 25 times less than that is required for CHIP. CAT-and-TAG takes only 1 to 2 days from cells to DNA library due to the time saving on in vitro adapt ligation. CAT-and-TAG also saves on sequencing costs because it requires less sequencing depth than CAT-and-RUN and CHIP assays. Like CAT-and-RUN, CAT-and-TAG can work with unfixed cells, helping to avoid cross-linking artifacts. CAT-and-TAG It is effective for histone modifications and certain transcription factors and cofactors. I will dive into more details about target versatility in my next slides. Similar to ChIP and Chiron, our CSC CAR-NTAG assay kit works with both rabbit and mouse primary antibodies because our TM5 is fused with both protein A and protein G. CAR-NTAG versus Chiron, which assay aligns better with your experiment's needs? Each coin has two sides. CAR-NTAG is not superior to Chiron in all aspects. I'd like to dive deeper into the differences between these two assays in terms of target versatility. The key enzyme used in the CAR-NTAG assay, the TM5 transposase, exhibits a binding bias towards open chromatin regions. To wash off the non-specific binding of TM5, a higher salt concentration is needed. In the coloration analysis, we observed that with a high salt concentration, about 70% of binding peaks of the H3K4 trimethyl antibody can match its ChIP-seq results. Chiron and the CAR-NTAG share a higher similarity compared to the ChIP-seq assay. With a titration of lower salt concentrations, we often observe more binding peaks. However, the match percentage between CAR-NTAG and the ChIP decreases, indicating the identification of more non-specific bindings. The correlation between CAR-NTAG and the other assays is poorer for chromatin repressors like H3K27 trimethyl, which are not expected to locate on open chromatin regions. And it's even worse for non-histone targets like CTCF. So now we face a dilemma. Lower salt concentration. With lower salt concentration, we can often see more kind of peaks. However, we will also see non-specific binding regions. With higher salt concentration, we will see more reliable peaks at the cost of losing more real binding but weak peaks. So what's the sweet spot? And this can be vary from target to target, from antibody to antibody. So we make a difficult decision to use high salt concentration in our kit and buffers to prioritize biologically meaningful signals. However, due to the high salt concentration in buffers, many weak bindings between non-histone targets and the chromatin DNA are not detected in CAR-NTAG. Consequently, this makes the CAR-NTAG assay incompatible with a broader range of non-histone targets compared to Chiron. When deciding between CAR-NTAG and Chiron for experiment, first consider the type of protein target you are interested in. If you are studying histone modifications, opt for CAR-NTAG due to its fast protocol compared to Chiron. For non-histone targets, choose CAR-NTAG if you can find a CAR-NTAG validated antibody. Otherwise, opt for Chiron assay. Secondly, think about how you plan to analyze your data. If you are focused solely on NGS, CAR-NTAG is the preferable choice because of its streamlined protocol. However, if you require qPCR data or both NGS and qPCR data, select Chiron since CAR-NTAG is not compatible with qPCR. Thirdly, consider the importance of time to your results. CAR-NTAG can save you a day for each round of experiment from cells to DNA library. If time is a critical factor, consider trying CAR-NTAG. Next, consider whether you anticipate conducting single-cell chromatin findings in the future. If so, CAR-NTAG is the only viable option. Lastly, think about whether you aim to pull more samples in a sequencing lane to save sequencing costs. If this is your goal, go with CAR-NTAG. So clearly, CAR-NTAG is a newer assay compared to Chiron, but that doesn't mean it should replace Chiron. you need to select the appropriate assay based on your experimental requirements. Next, let's discuss how CST ensures performance of all CAR-N kits, reagents, and antibodies to enable you to achieve consistent, reliable, and reproducible results. Each CST CAR-N assay kit supports 24 assays with a shelf life of 6 months. This kit comprises 4 degrees subassembly and a -20 subassembly. Proper storage of all reagents is crucial to ensure their functionality. In our experience, some customers occasionally install the entire kit at -20 or room temperature upon receiving it, without paying attention to the storage temperature. In such cases, if the whole assay kit is accidentally left at -20, please pay particular attention to the performance of the concatenate beads and the spin columns for DNA purification. If the entire kit is left at the room temperature for less than a week, the majority of the reagents are still good for use, including the PAT-TN5 enzyme and the concatenate beads. However, the spermidine solution and the proteinase inhibitor cocktail may have impaired performance. If necessary, all reagents in our CAR-N assay kit are available as standalone products. If you are following an in-house protocol, you also have the option to purchase 1 or more CAR-N reagents from this list for your convenience. Undoubtedly, one of the most valuable products in this list is the protein AG-fused TN5 enzyme. Expressing and purifying the TN5 enzyme are challenging because the enzyme is toxic to the host cell upon overexpression. CST's TN5 is fused with both protein A and protein G, optimizing its affinity for both mouse and rabbit antibodies. As demonstrated in the example with mouse and rabbit RBP1 antibodies, both antibodies exhibit strong and matching binding patterns on chromosome when used with CST-PAT-TN5 enzyme. The inclusion of this protein AG-TN5 in our CAR-N assay kit makes the entire kit ideal for use with both rabbit and mouse antibodies. The competitor comparison test demonstrates that CST-PAT-TN5 produces CAR-N signals comparable to those generated by other commercial sources of TN5. In addition to the reagents in the CAR-N assay kit, library prep reagents are required to prepare the CAR-N DNA library for NGS sequencing. The dual index primers and the PCR mask mix are unique to the CAR-N assay. Typically, the primer for library DNA amplification includes a reagent that is reverse complementary to the adapted sequences, allowing the primer and the DNA template to anneal in the PCR amplification reaction. However, the adapted sequence on CAR-N DNA is preloaded to TN5 and differs from the adapter ligated to ChIP and CAR-N DNA fragments in vitro. Therefore, you cannot use the dual index primers provided for ChIP and CAR-N library prep for CAR-N DNA library prep. Additionally, the ABP barcode sequences in CAR-N index primers are the same set of the barcode sequences used in ChIP and the CAR-N index primers. So although ChIP, CAR-N and the CAR-N samples can be put together for NGS, different barcode combinations should be used for each sample. For example, avoid using the same barcode combinations such as 701 and 501 for both CAR-N and the CAR-N samples, simply because the index primer used for each sample comes from different library prep kits. CST provides 96 barcode combinations, and typically less than 50 samples are put together in each sequencing run. So you should be able to find enough barcode combination for all of your samples in one sequencing run. Similarly, the CAR-N PCR mask mix differs from the PCR mask mix provided in our ChIP CAR-N library prep kit or the qPCR mask mix provided by CST. Here's why. When we examine the tegmented DNA structure at the end of the CAR-N experiment, there is a gap between adapter and the target DNA fragment. It's a feature of tegmentation. Therefore, we require a non-hot start DNA polymerase to facilitate the gap filling extension at 72 degrees for tegmented DNA. The PCR master mix in our ChIP and Chiron library prep kit is a hot start DNA polymerase, which is not activated until the temperature reaches 98 degrees. While it can help decrease some non-specific application during the temperature increase process, it won't work for the gap filling step, which requires polymerase activity to be initiated at a lower temperature. Therefore, if you prefer to design your own ChINTAC index polymerase, make sure to purchase our non-hot start PCR mass mix for your ChINTAC experiment, as CST offers it as a standalone product too. So overall, from cells to ChINTAC DNA library, each CST ChINTAC assay costs over 20% less than our competitors for each assay. As mentioned earlier, when deciding between the Chiron and the ChINTAC assays, one crucial factor to keep in mind is the availability of validated antibodies for each assay. Currently, CST provides the largest number of validated ChINTAC antibodies on the market, outpacing our competitors, especially when it comes to non-histone targets. It was noting that monoclonal antibodies, polyclonal antibodies, or different antibody clones targeting the same proteins are considered as separate antibody products. If we shift our focus to the number of validated ChINTAC targets rather than the total antibody counts, CST's validated ChINTAC targets significantly surpasses those of our competitors, demonstrating the extensive range and diversity of our offerings. Additionally, we have about 30 more non-histone CST targets validated for ChINTAC soon be available on our website. Looking forward, we are committed to continuously validating more ChINTAC antibodies to better serve our customers in the years to come. In validating ChINTAC antibodies, we implement a stricter set of criteria compared to ChIP and Chiron antibodies. This is because of the TM5 binding bias towards open chromatin regions. Naturally, CST's validated ChINTAC antibodies must meet our standard NGS pass-fail criteria, including considerations like peak numbers and the signal-to-noise ratio. However, we go a step further by cross-referencing the ChINTAC results of the working antibodies with their results from ChINTAC and Chiron and ChIP-seq experiments. This ensures that all identified ChINTAC peaks are not just statistically significant, but also biologically meaningful. For instance, during the validation of the SUS-12 antibody for ChINTAC, although it exhibited robust binding to the node target region, the HOX-A gene clusters, similar to its performance in the Chiron assay, it also displayed binding to the open chromatin regions around ACTB gene region, where such binding was not expected. The non-specific binding phenomenon was only observed in the ChINTAC assay, but not in the Chiron assay. Consequently, we deemed the SUS-12 antibody unsuitable for the ChINTAC application. After covering our range of reagents and antibodies, now let's shift our attention to our ChINTAC assay kit. I will walk you through some example data generated by our kit to give you a clear picture of its performance. I'd like to present some comparison data across ChIP, Chiron, and ChINTAC assays for all three target types, which have different challenging level in chromatin profiling assays. Let's kick off with histone PTM, which is the easiest target type to examine. Looking at the H3K27 trimethyl results, we observe a strong alignment among the ChINTAC, Chiron, and the ChIP-seq outcomes on the node genomic binding sites, particularly on the HOX-A and HOX-D gene clusters. You may notice that the ChINTAC signal tends to be stronger compared to the signals from Chiron and the ChIP-seq. This is actually true for transcription factors and cofactors as well. Now let's look at the second category, transcription factors. Unlike H3K27 trimethyl, which exhibits broad binding patterns on repressive genome regions, Transcription factors like ER-alpha display narrow binding peaks on specific gene regions. Once more, our CAR-Ntag assay kit demonstrates stronger signals compared to Chiron and ChIP. Whether you zooming out to view the entire chromosome or zooming in on promoter regions of its target gene, TFF1. Finally, let's tackle the most challenging category, cofactors. Here, we present the results from JERry-D2, which typically exhibits a broad binding pattern. As expected, the CST CAR-Ntag assay kit generates clear and robust signals on the HOX-A and HOX-D gene clusters with the JERry-D2 antibody. In addition to validating our CAR-Ntag assay kit across all protein target types, we've also assessed the minimum amount of starting materials required. Like our Chiron assay kit, only 5,000 cells are needed for histone targets. However, for non-histone targets like transcription ACTF or ATF4, the CAR-Ntag assay requires at least 20,000 cells. Nevertheless, it's still a significant reduction in material requirement compared to ChIP-seq, which typically needs 4 million cells for non-histone targets, which is 200 times more material. Moreover, we have validated our CAR-Ntag assay kit for use with various tissue types. Our findings demonstrate that it's efficient in detecting histone targets across different tissues, such as brain, heart, and liver. And here we present 2 examples showcasing both activator and repressor markers. Furthermore, we observe that the binding pattern of CAR-Ntag in tissues is similar as that seen in ChIP and Chiron assays. While we have achieved occasional success with non-histone targets in tissues, we have observed variations in optimal experimental conditions, such as initial amount of materials and fixation conditions on a case-by-case basis. Consequently, we do not recommend our standard CAR-Ntag protocol for non-histone targets in tissues. Instead, we recommend our Chiron assay kit for non-histones in tissues, as it offers a higher success rate for such experiments. Now, I'd like to present some data comparing our CAR-Ntag assay kit with those of competitors. While evaluating competitor CAR-Ntag assay kits, we adhere strictly to their provided protocols and utilize all supplied reagents, including index primers. So we are comparing both reagents and protocol from cell preparation to DNA library preparation. In the case of the histone target H3K27 trimethyl, the CST CAR-Ntag assay kit produces signal strength comparable to all 3 competitor kits when starting with 100,000 cells. Notably, our reagent yield a stronger signal than those of competitor 1 and competitor 2 when we use lower cell numbers. Similar results are observed with the non-histone target SMART-CC1, a cofactor in the BAF complex, using our CST-validated CAR-Ntag antibody. When starting with a high cell number of 100,000 cells, all 4 CAR-Ntag kits perform similarly. However, with only 20,000 or 10,000 starting cells, competitor 1 and competitor 2 show poorer performance compared to both CST and the competitor 3 kit. Furthermore, we conducted a comparison between our CST CAR-Ntag kit and a competitor's CAR-Ntag kit specifically designed for tissue samples. Our findings reveal that our CAR-Ntag assay kit produces comparable results for brain and liver tissues, which are relatively easier to work with. Interestingly, for heart tissues, which presents a more challenging fibrosis environment, our CST kit actually yields a stronger signal compared to the competitor. Finally, I'd like to provide some important insights into potential challenges you may face with the CAR-Ntag assay, along with resources it will provide to support your CAR-Ntag experiments. One challenge you may encounter with the CAT-NTAG assay is the yield of the DNA library. It is standard practice to measure chip and current DNA library yield and assess library quality on a bioanalyzer or tape station system before NGS. However, despite starting with a similar number of cells as in the current assay, the CAT-NTAG assay typically results in a lower DNA yield due to the higher specificity of the in vivo tegmentation reaction. This is also why the sequencing depth required for CAT-NTAG samples is less than that needed for CAT-NRAM DNA libraries, typically 2 million reads compared to 3 to 5 million reads, as mentioned previously. The yield of the CAT-NTAG DNA library may be so minimal that it fails to generate a signal on a bioanalyzer or tape station system. However, as demonstrated in previous slides, CAT-NTAG DNA libraries can often produce stronger signals than CAT-NRAM and chip libraries, even with less DNA loaded onto the sequencing machine. Therefore, please do not apply the same expectations derived from the chip or CAT-NRAM DNA libraries to CAT-NTAG DNA libraries, because a low yield CAT-NTAG DNA library can still produce quality data. For instance, in the example data provided here, both CSTs and the two competitor TM5 enzymes generated very weak or even invisible peaks on the bioanalyzer with the CAT-NTAG DNA library using a transcription factor antibody TCF4. However, all of these three low-yield CAT-NTAG DNA libraries produce decent binding peaks in the NGS results. If you require further guidance on how to proceed with NGS using low-yield CAT-NTAG DNA libraries, you can refer to our CAT-NTAG FAQ webpage and blogs. For example, we employ a formula to convert library concentration from nanogram per microliter to nanomole before diluting each library sample to the same concentration in nanomole for pooling purposes. Typically, the software in the bioanalyzer or tape station will provide the average size of the library DNA pool, which you can use in this formula. However, in case of flat peaks, we recommend using 900 base pair, the largest size of DNA fragment compatible with NGS, to intentionally pool more low-yield libraries than normal yield libraries. Furthermore, when pulling together samples with a normal bioanalyzer peak and those with a flat bioanalyzer peak, we suggest pulling the libraries with a flat signal 5 to 10 times more than those with normal-sized peaks on the bioanalyzer or tape station systems. This helps achieve an even distribution of the number of reads among all samples. When the bioanalyzer or tape station isn't suitable for QC of CAT-NTAG samples with flat peaks, there are alternative methods to predict whether CAT-NTAG DNA library can deliver quality NGS data. Firstly, we recommend running a positive control like H3K4 trimethyl alongside your DNA library to assess protocol success. If the positive control signal is strong on the bioanalyzer or tape station system, it indicates that the reagents and the protocol workflow are functioning effectively. In such cases, even if the signal from the target library is low, the CAT-NTAG DNA library can still be sequenced to provide valuable DNA protein interaction data. Secondly, although CAT-NTAG DNA isn't compatible with qPCR because the sample is composed of both tegmented DNA and background genomic DNA, qPCR analysis of target genes is still feasible using a CAT-NTAG DNA library. During the library application process, the tegmented DNA fragments are selectively enriched while the background genomic DNA is diluted out. So here is an example. The blue bar and orange bar present the positive gene loci, while the gray bar represent a negative gene locus. Prior to library prep, qPCR on CAT-NTAG DNA exhibits high background and a low signal-to-noise ratio. However, after library preparation, qPCR on the CAR-NTAG DNA library demonstrates a significant signal-to-noise ratio, indicating a successful CAR-NTAG experiment. Here are some common causes if low sequencing depth or no NGS signal is observed in your CAR-NTAG experiment. Firstly, ensure that your assay started with an adequate number of cells. Inaccurate cell count, cell loss analysis may result in fewer cells being used in the assay. Minimize cell loss by performing initial cell wash steps in one vial. The number of cells required for optimal enrichment depends on the abundance of the target protein and the affinity of antibody. Ensure that the starting cell culture is 60 to 90% confident and appears healthy, with over 90% of live cells. Avoid treating cells with trypsin for longer than necessary. Prepare cells at room temperature and deal with it as quickly as possible to minimize cell stress. Secondly, use live cells or fresh tissues for CAR-NTAG assay whenever possible. If live cells or fresh tissues are unavailable, lightly fix the cell tissues for 2 minutes in 0.1% formaldehyde. Overfixation can inhibit the tegmentation reaction and result in signal loss. Thirdly, ensure that digitoning effectively permeabilizes cells. If needed, test and confirm the optimal amount of digitoning following our appendix protocol. Always thaw digitoning solution completely before use because it may crash out of solution after being placed on ice for a while. Next, verify the performance of your antibody. Not all antibodies are suitable for use in CAR-NTAG, especially those targeting non-histone targets. Whenever possible, use CAR-NTAG validated antibodies. Include a positive control antibody to confirm the overall experimental success. The fifth possibility is that the PAG-TM5 enzyme is not functioning properly in the assay. Do not use expired PAG-TM5 as its activity declines over time. Store the PAG-TM5 enzyme properly at -20 with a shelf life of 6 months. If the DNA library yield is very low, consider increasing the number of PCR cycles to amplify tegmented DNA. The optimal number of PCR cycles depends on the starting cell number, typically ranging from 13 to 16 cycles. You can refer to our protocol for CAR-NTAG dual index primers and PCR mass mix for Illumina, even if you're not using our library prep reagents. For further information and recommendations regarding the CAR-NTAG assay, please visit our CAR-NTAG Resource Center, now conveniently accessible on our CST homepage. This resource hub contains links to the CAR-NTAG overview, CAR-NTAG troubleshooting guide, CAR-NTAG FAQ, CAR-NTAG protocols, and a dedicated CAR-NTAG blog. You will find a range of topics covered, including fixed cells preparation, fixed tissue preparation, determination of cell sensitivity to digitoning, and more. So here is the key takeaways from today's webinar. CAR-NTAG uses target-specific primary antibodies and PAG-TM5 to isolate protein DNA complexes and ligate adapt DNA in vivo. Benefits of CAR-NTAG include low sample requirement, fast time to results, sequencing cost savings, avoidance of cross-linking artifacts, target versatility, and antibody versatility. Factors to consider when choosing between CAR-NTAG and CAR-RUN. First, look at the protein targets to be analyzed. Is it a histone mods, transcription factors, or cofactors? Then consider the model system. Is that cells or tissues? Consider type of analysis to be performed, qPCR or NGS. The CST CAR-NTAG assay kit is validated for use with all 3 protein classes in cells and for use with histones, PTMs, in tissues. CST offers the most validated kit, antibodies, and the standalone reagents for CAR-NTAG. Thank you for your attention. All the resources I mentioned for the CAR-NTAG assay can be found on this webpage or by scanning this barcode. If you have any questions regarding antibody selections or assay troubleshooting, please feel free to reach out to us by emailing support@cellsignal.com. If for any reasons you determine that cut and run is a more suitable assay for a target, CST also provide cut and run products for your convenience. Now I'm glad to answer any questions you may have.
Thank you for that great presentation, Dr. Chen. I see some of you have already put questions in, so as a reminder, if you have any questions you'd like Dr. Chen to address, please put them in the questions box on your GoToWebinar control panel, and we will answer as many as we can today. Dr. Chen, to start off, there's a couple of questions about the cut and tag kit, and does it work for frozen tissue or FFPE tissue? Will it work on tumors or peripheral PBMCs?
Yes, our cut and tag assay kit works with fresh frozen tissue samples or tissue samples directly frozen at -80. So after thawing, fixation of the tissue sample is actually optional, as we haven't found that fixation boosts the cut and tag signal strength. As for the FFP tissue, we haven't got a chance to test our kit with FFP tissue yet. But I can recommend a 2023 Nature Communication paper from Dr. Henikoff's lab, which introduces a method to reverse cross-link the FFP tissue for cut tag. And I think this protocol should also work for cut and tag tissue samples pre-treatment. I can share that paper after this webinar. Excellent.
Would there be any changes for PBMCs?
No, we don't have experience on that. Sorry for that.
Okay, excellent. There are a couple of questions about controls, and can we use spike-in DNA in cut and tag for sample normalization? And another customer wants to know if they spike in orthogonal chromatin from another species, where in the protocol would they do that? So is it possible, and how would we do it?
Well, in our cut and run assay kit, we do provide the spike-in DNA, but unfortunately, the spike-in DNA is not compatible with cut and tag. That's because tegmentation, takes place within the nuclei. To minimize the non-specific background, and excess TM5 enzyme is washed away prior to activation. And similarly, spike-in DNA introduced to the reaction would be eliminated alongside with excess TM5 if added before tegmentation initiation. And once tegmentation is complete, there's no benefit in adding spike-in DNA to the samples. This is because the spike-in DNA doesn't undergo tegmentation alongside antibody-targeted genomic DNA fragments. As a result, during the PCR amplification of the cut and tag DNA library, the spike-in DNA would be lost due to the absence of the adapter sequences on its ends. So consequently, spike-in DNA is not compatible with the cut and tag assay. The customer had a very good question. I think yes. So if they incorporate an equal amount of pre-isolated nuclei from another genome along with an antibody specifically target that genome in the testing reactions, it can serve as a spike-in for sample normalization purpose. Or alternatively, you may consider choosing cut and run assay, which offers option for spike-in DNA.
Okay. Can you sequence cut and tag DNA with MiSeq?
Yeah, that depends on the number of samples you have and the sequence depths you desire, right? MiSeq, if I remember correctly, provides 25 million reads for each sequencing run, and HiSeq provides 400 million reads for each sequencing run. So as I mentioned that for cut and tag samples, we recommend 2 million reads per sample. So just count like how many. Maybe I think for MiSeq 25 million, maybe you can pull up to 10 samples. That would be safe.
Okay. Someone wants to know how to understand whether an antibody is compatible for cut and tag, and is there any checklist to go through before their experiment?
Yes, CST on CST website, if you search cut and tag antibodies, like you will see the list of all of the CST validated CAR-N antibodies. So far, we have 33 primary antibodies and 2 secondary antibodies. And as I mentioned, we do have 30 more antibodies already validated in-house. We are now in the middle of the paperwork to release them on web. So they will be there very soon. We will have like over 60 CAR-N validated antibodies for you to choose.
Okay. I'm going to add that if you're ever unsure, you can reach out to support@cellsignal.com or your local account manager, and they'll help you figure out whether an antibody's been tested or is in the process of being tested.
Exactly. If you don't find it, if you see the antibody but didn't say it's CAR-N-validated or not, feel free to reach out to us to see, well, it's tested but failed or it's been tested yet. That will give you information. Yeah.
Do you recommend DNA size selection, ampere beads, etc., prior to library preparation?
No, we never recommend size selection because, especially for CAR-N DNA sample, the library yield is so low. Every single DNA molecule is so precious. So any kind of purification will cause like 50 to 80% of 20 to 50% of loss of the sample. So we don't recommend that. Yeah, no. The answer is no.
Okay. Is the DNA cleanup column kit already included in the cut and tag kit and not required as an additional purchase, as it is not included in the cut and run kit?
Right, DNA purification columns is included in our cut and tag kit for your convenience. The reason we didn't include that in cut and run kit is because many cut and run users, they still prefer to use the phenylchromophore to purify the small cut and run DNA. But for cut and tag, you know, all of the DNA of interest you already have adapted ligated. So the DNA size is actually at least over 70 BP, and it's completely compatible with the spin columns. Our spin columns can recycle DNA over 35 BP, but if you have any concern, now it's like 70 BP, definitely the spin column can do the work. And it just, I don't think it worth the message to do phenylchromophore for a relatively large DNA fragments.
Okay. Do you have a combination chart of compatible primer mixes when using common combinations, such as 6, 12, 18, 24 samples?
I don't get that question. So when you say the common combinations, actually there's no any preference, like there's no preference on the number of the samples to put in one sequencing run. It's purely based on your needs. In our in-house test, we may pull like 23, 47, you know, it's just how many samples we have, we just put them together. No, we don't have to stick with a certain number.
Okay. Lots of questions, which is great. How does the cut and tag assay perform with cells containing high lipid content?
That's a good question. So for a special cell type like fat cells or muscle cells, you know, these are all special cell types. We would recommend some pre-treatment on the cells using specific protocol before starting the experiment. So I think it's same for cut and run and the chip as well. Yeah.
Okay, a couple more questions about working with tissue. How do CST freeze fresh tissue? Is flash frozen prohibited? And then somebody else wants to know, is it markedly better to use fresh or frozen tissues with cut and run? And does it work with brain tissue?
Well, we compared fresh frozen tissue. We put the tissue in liquid nitrogen for 2 minutes and then, you know, start at -20, or we just directly start the tissue to -80, and then we compared. We don't see a difference between different frozen methods. So any freezing method works. So my point is that the fixation after sawing is probably optional. Because we tried fix saw and then fix saw without fix. no signal difference. So I don't feel like the fixation really worth the time.
Okay. Do you see any difference in fresh or frozen tissue with cut and run? And have we ever tried it with brain samples?
Fresh tissue is always the best. Well, to be honest, in our in-house test with brain tissues, we use a whole brain. It's not like small, section of specific brain cell type. So with whole brain, works totally fine.
Okay.
With whole brain is a very easy sample tissue to deal with. Yeah.
Okay, so more sample questions. I've noticed there are several cut and tag protocols that start with nuclei extraction. Does your kit work with isolated nuclei, and does isolated nuclei yield better results?
Yes, our kit works with both whole cells and the pre-isolated nuclei because the concatenate beads bind to glycoproteins on the surface of both cells and nuclei. However, our internal testing data shows that pre-isolation of cell nuclei doesn't help to increase the cut and tag signal. So pre-isolation of cell nuclei should only be necessary for cells that are not effectively cannibalized by digitoning, such as yeast or plant cells, which both contain cell walls. It's doable, but not necessary.
Okay. And then I think somewhat related, does the kit work for single cell cut and tag?
It should. We haven't validated our cut and tag assay kit with single cell assays yet, but I know at least 3 publications that perform bulk cut and tag assay first, and then like it with a 10x genomics assay kit to isolate the single nuclei for a PCR in each nuclei. So it's definitely doable, and it's very, very easy. Just at the end of the experiment, you just change the buffer to 10x genomics buffer and follow their protocol to continue. It's definitely doable, and I'm happy to share these publications if the customer is interested. Yeah.
Okay, excellent. Follow up on finding the right antibody. What if I need to look at a target for which there are no validated antibodies for cut and tag?
Well, if no, I then recommend try cut and run assay or chip assay. But if you really want to do cut and tag assay, you may try cut and run validated antibody first, and then try chip-seq validated antibody, and then try IF validated antibody. But I would like to say cut and run validated antibody may have like 70–80% of success rate in cut and tag, but for chip and the IF, maybe 50% of the success rate to work in the cut and tag. Yeah, that's my intention.
Okay. So if the antibody hasn't yet been tested for cut and run, or for cut and tag, cut and run validation provides a 70–80% chance generally that it might work, and then chip and IF validation, less of a correlation, but still perhaps it will work.
Yes, yes.
Is there an easy way to check if your cut and tag assay works without sequencing?
Without sequencing, yes. As I mentioned in my slide, you can run PCR on cut and tag DNA library. So DNA library is very easy to make just one more PCR amplification, and that library DNA sample is ready for qPCR, and then you can quickly check the signal-to-noise ratio. Don't forget to include the input sample in your experiment, because you will normalize your PCR signal back to input sample. Then that's it. Yeah, PCR is doable on library DNA.
Okay. Can we study RNA interactions with DNA with cut and tag?
Probably not. I'm not sure if tag augmentation happens on RNA, and I don't know how to differentiate RNA and the DNA portions at the end of the experiment. I don't think cut and tag is a proper assay for RNA DNA interactions.
Okay.
Yeah.
So not the right method. Have we done any other cut and tag profiling on brain samples besides histone H3 trimethylhistone H3 lysine 4.
Yes, we have done many different histone targets, and they all work very well in brain. We also tried several non-histones actually in tissue samples, including brain. Some of them work, some of them not. And then for the working ones, they only work for like a certain starting material or a certain fixation condition or whatever. So we feel it's very hard to conclude a consistent general protocol for non-histone targets. So that's why we don't recommend CutNTag for non-histones in tissue samples, because it takes too long for you to optimize the protocol. Maybe just to cut around, that would be easier and more efficient.
Okay. Somebody else is looking at histones and says, if I'm looking for histone modifications in Drosophila tissue with qPCR as the endpoint, would cut and run or CutNTag be a better choice?
If I think cut and run would be a better choice, because CutNTag is a little bit overkill, right? You don't need that library, but you prepare the library. So maybe just to cut and run, you can directly use cut and run DNA for qPCR. So that's actually slightly easier.
Okay, yeah. So cut and run would be the best choice there for that type of experiment.
Yeah.
Can you use the CST kit with a non-CST antibody that has been validated for CutNTag?
That's a very good question. Yes, you can, but we cannot guarantee the results because it's unvalidated antibody.
All right. Do you need an input sample for CutNTag sequencing?
That's a good question too. So as to the negative control for the CutNTag experiment, we actually have two different controls. One is input control, one is IgG control, and each of them have their own advantage and the limitations. So the input DNA sample is fragmented genomic DNA that's not segmented by TN5. So it requires additional bench work compared to the CutNTag samples from the same experiment, including sonication fragmentation and the in vitro adaptive ligation steps for library prep for input. But the benefit of input DNA sample is that it always generates enough library yield and the sequencing reads for data analysis. Using our own peak calling algorithm, some background signal can end up being counted as peaks if the input DNA sample is used as negative control. And an IgG control performs better with regard to reasonable and reliable peak calling results. However, an IgG control has a risk to obtain a lower library yield with significantly fewer sequencing reads for the data analysis. Usually, sometimes only get 500,000 reads or even less reads, not enough for data analysis. So which control to use is really up to you and up to your bioinformatics algorithms. So we know that some labs analyze CutNTag peaks without any negative controls. But that said, we still provide both rabbit and mouse IgG antibodies in our CutNTag kit for convenience. And the input DNA prep protocol is also available on CST webpage if you're interested.
Okay. And I think this will be our last question for today. Is CutNTag compatible with nuclei that have been pre-extracted for other preparations like ATAC-seq?
It depends on, as I said, CutNTag works for nuclei, right? But really depends on if the nuclear sample, how is it fixed? So we want light fixation on the samples. So if it's like overfixed, like we want 0.1% for metahypertomies. If it's overfixed than that, then it's not compatible. If it's lightly fixed, or then it should be usable.
Okay, great. Well, our time is up. We appreciate you attending our CutNTag webinar today and participating in the Q&A. If we were unable to answer your question at this point, we will be following up with you directly via email. Everyone, please be on the lookout for our post-webinar email. It will be sent out no later than June 20th and will contain the promo code for 25% off our CutNTag assay kit and index primers and master mix kit. Thank you again and enjoy the rest of your day.
Thank you, everyone. Have a great day.